The next lecture in our carbohydrate series is focused on glycolysis. So referencing back to our integrated picture of carbohydrate metabolism, we're now going to focus on this bit right here. So this is the conversion of glucose to pyruvate via glycolysis. We'll also look at the aerobic and anaerobic scenarios as endpoints for glycolysis. And we'll also cover how your cells dispose of the glycolytically produced NADH, which is also another source of ATP. But before we start, I just want to go through some, I guess you'd call them biochemical definitions of particular terms. You see, biochemistry is like any discipline. There's a certain amount of terminology or language that goes with it. And if you get your head around some of those basic terms, it can really help your understanding of the rest of the material. It's almost like being able to see through someone else's coded language, if you like. And all of a sudden, things can seem a great deal simpler. So here's just a few definitions that if you commit them to memory, they'll make the rest of this material heaps easier to comprehend. First of all, some enzyme names. You can see three of the key ones here that I want you to commit to memory. Firstly, we've got a kinase. So for example, there's an enzyme called protein kinase. Now a kinase is any enzyme that adds a phosphate group using ATP as the phosphate donor. So this enzyme will add its phosphate group to its target substrate. Now the next one down is called phosphorylase. So a phosphorylase is any enzyme that adds a phosphate group, but in this case it uses an inorganic phosphate as the phosphate donor. So in some ways it's doing the same thing as the previous enzyme, that kinase, except in this case it's not using up ATP to do that phosphorylation. And a great example, which you'll hear about in the coming lectures, is an enzyme called glycogen phosphorylase. And the last enzyme is called a phosphatase. So this is any enzyme that removes a phosphate group and it frees it up as inorganic phosphate. So for example, protein phosphatase is one of these types of enzymes. Okay, so that's three specific enzyme names, and you'll see them used quite frequently through many of these pathways. So there's the kinase, the phosphorylase, and a phosphatase. Now the next thing I want you to commit to memory are some of the key ways in which we can regulate the activity of enzymes within metabolic pathways. There are four that I want to show you, and you can see them listed here. So the first is very simple. It's just substrate or product stimulation or inhibition. So this is an example of where just simply the presence of an enzyme substrate or the presence of its product will either speed it up or slow it down. Now the next controlling mechanism is through the enzymes Km and its Vmax. An enzyme with a very low Km and therefore high affinity for its substrate will also generally have a very low Vmax. And likewise, an enzyme with a very high Km, or if you like, low affinity for its substrate, will tend to have a very high Vmax. And we've already talked about some really good examples of this form of regulation when we were discussing the GLUT transporters. So GLUT 1 and 3 had a very low Km, so they could operate at very low substrate concentrations of glucose, but they also have a relatively low Vmax. By contrast, GLUT 2, the one that was on your liver, that had a very high Km, so it would only operate when concentrations of glucose were really high. And correspondingly, it's also got a really high Vmax. Okay, now our third form of regulation is through allosteric effectors. You can see here I've given you a picture of an enzyme, and you can see the active site that it will act on the substrate through. An allosteric effector will bind to that enzyme at somewhere other than its active site, and you call that the allosteric site. So for example, cyclic AMP is a classic example of an allosteric effector. It will bind to an enzyme, changing the shape of its active site, and in some cases speeding it up, and in some cases slowing it down. It really depends on that specific scenario. But nonetheless, this is called an allosteric form of regulation. And our last form of regulation is covalent modulation. So this is where an enzyme is phosphorylated, and by phosphorylating it, you will either speed up that enzyme or again, you will slow it down. Now classically, it is kinases that will cause the phosphorylation of a target enzyme, regulating its activity. And then correspondingly, it's a phosphatase that will come along and remove that phosphate group and then again reverse that effect. And I'm being careful here not to say that it's speeding it up or slowing it down. Again, it depends upon the scenario. So this is covalent modulation, where the phosphorylation of an enzyme will change its activity. So I can fully advise, take your time to get across those definitions across these last two slides. If you commit those to memory, it'll really make it easier to understand the rest of the material that I'll be describing in biochemistry. These mechanisms are also broadly quite crucial to understand. Many drugs that will be administered to animals will affect them through effectors or through potentially knock-on covalent modulation effects. And also many diseases will manifest through these mechanisms. Alright, so let's now dive in and have a close look at glycolysis, in particular looking at how it's regulated. A couple of high-level statements to start with. First of all, this pathway is the major pathway for carbohydrate oxidation. It breaks down carbohydrates as glucose to produce energy in the form of ATP, and the reactions proceed through a series of phosphorylated intermediates, ultimately ending up producing pyruvate. Now, all of this occurs in the soluble cytoplasm, or the cytosol of your cells, and all cells in your body will undertake glycolysis. In fact, glycolysis is thought to be the most ancient of all metabolic pathways. And quite simply, this is because it is present in all known living cells. And that's actually a fairly good argument for the existence of evolution, given that all cells have this pathway in common. As a general theme, the cells in our bodies and the bodies of animals become more dependent on glycolysis to produce energy under particular scenarios. These include when there is high levels of glucose intake, under high levels of stress, and particularly under high levels of exercise. But having said that, there are tissues in your body that are quite dependent upon glycolysis for their energy production. And they include the ones that you can see here in this list. So the brain's a crucial one, but a number of other cells really rely on it because they don't express mitochondria. And of course, it's within mitochondria that the aerobic pathway occurs. That's the TCA cycle. So if you don't have mitochondria, you become far more reliant, or in fact entirely reliant, on glycolysis for your ATP production. So good examples of cells without mitochondria include your red blood cells, so the erythrocytes. While these other ones here, so type 2 muscle, the fetus, the renal medulla, the retina and your skin, they do have some level of mitochondrial expression, but just at much lower levels. So generally, they're far more dependent upon glycolysis for energy production as well. So let's have a close look at the pathway. This is it here, and you can see that it consists of a series of 10 enzymatically catalyzed steps, starting with glucose and finishing with pyruvate. And all the way through here you can see the names of the enzymes in these grey boxes that catalyse these reactions, and also the names of the substrates. Now, back in the day when I was learning biochemistry, they actually made me learn this. It was quite hideous. And do you think I could remember all of this these days? Well, the truth is I actually can, but that's only because I teach it. It doesn't really do me that much good to know all of this micro detail. In fact, back in the day, they used to get really excited and try and make us learn all of the substrates that were involved, which you can see in this picture here. I'm kind of thinking maybe that was because it hadn't been discovered all that long ago. But nonetheless, definitely do not learn this level of detail. It won't help. So what I'm now going to show you is precisely the level of detail that I want you to understand about glycolysis. And most importantly, that includes the rate limiting steps and how they're regulated. You also need to know at a reasonably high level the basic substrate structures that are involved and also the energy yield from the different phases of glycolysis. So let's have a look at that. Here's the overall pathway as a starting point. This consists of, in the first steps, glucose and therefore 6 carbon substrates through this early phase here. And then partway down, this 6 carbon molecule gets split by aldolase into 2 3 carbon molecules. And so therefore the bottom half of this pathway is dealing with the gradual oxidation of 3 carbon molecules all the way through to pyruvate. So we transition into these 3 carbon molecules at this aldolase step right here, where we effectively chop the 6 carbon molecule in half. Now, in terms of the energy yield, there are effectively 2 phases involved. The first is like an investment phase that occurs up here. This actually costs energy in the form of 1-ATP when we initially phosphorylate glucose and then again phosphorylate fructose 6-phosphate. And if you have a look at the names of the enzymes that catalyze these 2 steps, you'll see that they're both kinases. Remember, that's one of the enzymes that I introduced earlier. But the main message here is that we invest 2 ATP to process glucose through this first investment phase. And then, in the second half of glycolysis, we generate 2 ATP, one here and one down here. But the reason why I've written a ×2 sign here and here is because we're dealing with 2 3 carbon molecules. So you remember our 6 carbon molecule got cleaved back here at the aldolase step. So we get to do this bit twice. And therefore the total energy yield is minus 2 ATP from up here in the investment phase plus 4 ATP, giving us a total energy yield from 1 molecule of glucose catabolized through glycolysis of 2 ATP. Alright, so that was the energy bit. Now let's get into the important stuff. Let's look at how this pathway is regulated. And to regulate this pathway, we do so through the 3 steps that you can see here, catalyzed by hexakinase, phosphofructokinase 1 or PFK1, and pyruvate kinase. I've coloured their arrows in red, and that's because they are one-directional reactions or physiologically irreversible. And these steps are also allosterically regulated. Now, can you remember what allosteric regulation was? That's where you've got an effector molecule that binds to an enzyme to affect its rate, and it binds somewhere other than its active site. So the allosteric regulation is occurring at these physiologically irreversible steps, and that makes really quite good sense. Okay, so let's have a look at what regulates those 3 steps, starting with hexakinase. Now the key job of hexakinase is to phosphorylate glucose, and by phosphorylating it, you anchor it within the cell. So this phosphate group effectively means that glucose is not permeable back through the cell wall or out through those GLUT transporters. So this is a very crucial first step. It's effectively locking glucose inside the cell so that the cell can use it. Now, in part, hexakinase is controlled allosterically by its product, glucose 6-phosphate, which of course slows it down. However, its principal point of regulation is through its Km. And you can see here that its Km is 20 micromolar. So that's a really low value. particularly if you consider homeostatic levels of glucose concentration in your blood, which are sitting at 5 millimolar, so far more concentrated than that. So this means that because of that really low Km and therefore that really high affinity for glucose, hexokinase will always be able to find its substrate and phosphorylate it. But the downside of this is that by having such a low Km, you'll also have a really low Vmax. Now, generally, this isn't too big a drama unless you've just had a big feed of carbohydrate. So you've got glucose flooding into your bloodstream. And this is crucially important in your liver. Your liver's principal job is sitting there like a big sponge on the end of your portal vein, draining your gut. So when huge amounts of glucose are being absorbed from your gut, your liver sits there and filters a good portion of it out. It's got those really high Km and therefore really high Vmax GLUT2 transporters. Now, can anyone remember what the Km of the GLUT2 transporters was? I'll let you think about that just for a second. Yep, the GLUT2s were sitting at a Km of somewhere between 12 and 20 millimolar of glucose. So that means they've got a massive capacity to absorb glucose into the liver cells, particularly when substrate concentrations are really high. And that's quite a mismatch for the cell's ability to then phosphorylate that glucose so that it doesn't simply flux back out. Well, fortunately, your liver has the answer. Not only does it express GLUT2 transporters, but it also expresses an enzyme called glucokinase. Now, glucokinase does the same job as hexokinase in that it phosphorylates glucose as it enters your liver cells. But the difference here is that it's highly specific for glucose, and in addition to that, have a look at its Km. It's got a value sitting up at 10 millimolar, which is a far better match for those GLUT2 transporters that had a Km sitting at 12 millimolar. So therefore, after a high carbohydrate meal, where you've got glucose flooding into your bloodstream, your liver, equipped with GLUT2 transporters, can absorb a good portion of that glucose out of the blood and then phosphorylate it using glucokinase, effectively locking that glucose inside the liver cell. And the other nice thing about glucokinase is that it is not inhibited by glucose 6-phosphate, which is, of course, its substrate that it produces. So therefore, there is no end product inhibition of that enzyme. So again, that's just another nice story of how your body has adapted the enzymes that it uses through their Km to regulate particular pathways. As a side note, can you remember how I was describing earlier that ruminants don't absorb much glucose directly from their diet? And that's because when it enters their rumen, it gets fermented and broken down into volatile fatty acids. So therefore, ruminants, and in fact horses, because they don't have that huge capacity to absorb glucose, they're rarely flooding their livers with it. And on that basis, they don't need that glucokinase adaptation. So what you find is that the liver of horses and ruminants don't express glucokinase. This is very much a monogastric mechanism. All right, moving along down the pathway, the next regulatory step that I want to tell you about is this one here, phosphofructokinase. Phosphofructokinase, or PFK1, is the most important regulatory step of glycolysis. And the reason why is because it's the slowest. It's regulated allosterically, but there are a number of mechanisms that can impact. It can be both inhibited and stimulated through different allosteric effectors. ATP, citrate, and hydrogen ions can all act to inhibit this enzyme, and AMP and fructose 2,6-bisphosphate can act as stimulators of this enzyme. Now, I'm going to particularly focus on this allosteric effector here, fructose 2,6-bisphosphate. And that's because it's the most potent effector. If this stuff here is present, it'll swamp the effects of all of these other allosteric effectors. So when fructose 2,6-bisphosphate is produced, it has a massive stimulating effect on PFK1, or phosphofructokinase 1. And it's made from a glycolytic substrate, but don't be fooled, it itself is not a glycolytic substrate. And it gets made by a bifunctional enzyme. Now this enzyme has two halves. There is one half called PFK2, or phosphofructokinase 2, and its job is to make fructose 2,6-bisphosphate. The other half of this bifunctional enzyme is called fructose 2,6-bisphosphatase, and its job is to break down this allosteric effector. So therefore, to speed up glycolysis, we need to make more of this allosteric effector, fructose 2,6-bisphosphate. And therefore, we need the PFK2 half of our bifunctional enzyme to be active, making the allosteric effector. But if we want to slow glycolysis down again, we need to remove this allosteric effector. And we do that by activating the degradative path of this bifunctional enzyme, fructose 2,6-bisphosphatase. So the crucial thing is to control the rate of activities of the two halves of this enzyme. Now, I won't tell you too much about that just at the moment. In a couple of lectures' time, we'll get into gluconeogenesis. And I'll show you in that lecture how we control the two halves of this enzyme and regulate its activity. But nonetheless, the effect of fructose 2,6-bisphosphate on PFK1 is clear. It massively increases the affinity of that enzyme for its substrate without diminishing its Vmax. And you can see the contrast in black of that same enzyme when there is no fructose 2,6-bisphosphate present. Alright, so now let's look at our last regulatory step of glycolysis, catalyzed by pyruvate kinase. In this case, this enzyme is inhibited by ATP and also by glucagon when you're starving. This is actually one of the mechanisms during starvation that helps you preserve glucose. And in contrast, when you're well fed and there's lots of glucose in the system, you get a stimulation of pyruvate kinase via an allosteric feed-forward mechanism. So lots of glucose flooding in means there's lots of fructose 1,6-bisphosphate, which is a glycolytic substrate, feeding forward, stimulating the pyruvate kinase step of glycolysis. So we've now covered all of the regulatory steps of glycolysis. But what we haven't dealt with is how we dispose of the glycolytically produced NADH. And this brings about the concept of lactate being a final electron acceptor. Here's the step that I'm referring to right here, in which NAD+ is converted into NADH. And this is catalyzed by the dehydrogenase enzyme that you can see here, glyceraldehyde 3-phosphate dehydrogenase. And the problem with this scenario is that your cells only have a very limited supply of NAD+. It's a bit like having a very small cup that's easily emptied. So for glycolysis to proceed, it's crucial that we continuously recycle this NADH back to NAD+. Its role here was to pick up an electron from glyceraldehyde 3-phosphate. So we need to find an alternative electron acceptor, enabling us to recycle this molecule. And this is where lactate comes in. Through the activity of lactate dehydrogenase, pyruvate is converted into lactate, accepting an electron from NADH and effectively converting it back to NAD+. And on this basis, NAD+ is recycled and can continue potentiating glycolysis. And this is precisely what happened in an anaerobic scenario. I'm sure everyone here has heard of lactic acid being produced when you sprint or do high-intensity exercise. High-intensity exercise well exceeds the capacity of aerobic production of ATP. And yet glycolysis, and therefore the anaerobic production of ATP, can quite easily keep up, having a massive capacity to accelerate. But in doing so, your cells are forced to produce lactic acid so that it can keep recycling the NAD+, potentiating further glycolysis. Now, if this goes on for too long, the buildup of acid within your cells can ultimately become inhibitory, both to glycolysis and also to contraction itself. But this is definitely a mechanism to cover a short-term gap in energy. The enzyme lactate dehydrogenase, which forms lactic acid, exists as a four-part structure. So effectively it's got four catalytic subunits. These can be either the M form, which converts pyruvate to lactic acid, or the H form, which converts lactic acid back to pyruvate. But in most tissues, it's a mixture of these two isoforms. So in effect, lactate dehydrogenase can catalyze this reaction in both directions. The liver is a classic example of this. Alright, so that was the anaerobic scenario for glycolysis. I'm now going to tell you about the aerobic scenario. And in this case, oxygen acts as the final electron acceptor. Once again, the crucial thing is to recycle the glycolytically produced NADH. And this is done by shuttling it into the mitochondria through these electron shuttle structures and then coupling it with oxidative phosphorylation to form ATP. In this lecture, I won't tell you about the detail of how this works. I'm going to save that up for the next lecture. But for now, you can be rest assured that this enables the recycling of NADH back to NAD+, so that glycolysis can keep going, even in an aerobic scenario. So in this situation, glucose is converted all the way down to pyruvate, and pyruvate can be converted into acetyl-CoA and then fed into the aerobic TCA cycle, ultimately producing even more ATP in here. Whilst this process is far more efficient in terms of ATP yield, it can't be sped up anywhere near as much as pure anaerobic glycolysis all on its own. To finish with, I need to tell you a bit about the regulation of this enzyme here, pyruvate dehydrogenase. This enzyme is not strictly speaking a glycolytic enzyme. However, it is crucial to the final metabolism of glucose carbon. Pyruvate dehydrogenase is quite a complex enzyme. It consists of three catalytic enzymes or subunits, but it also consists of two regulatory enzymes. So these actually regulate its own activity. This enzyme is located in the mitochondrial wall and it converts pyruvate and NAD+ into acetyl-CoA, NADH, and a bit of CO2 and hydrogen ions. But there is one thing that is absolutely crucial to understand about this enzyme. It is physiologically irreversible. So this means that once pyruvate has been converted into acetyl-CoA, you can never get this carbon back into a form that you can recreate glucose from endogenously. Now, that may not sound like such a big deal, but if you're starving, then the most crucial thing in your body is to produce glucose to feed your brain. If you can't do this, you'll die. So on this basis, it's quite a big decision as to whether your cells will convert pyruvate into acetyl-CoA, passing through this irreversible step. So ultimately, the regulation of this enzyme is completely linked to energy supply and demand of the cell. So can you guess when you're likely to shut this enzyme down? Yep, you guessed it, it's when you're starving. So let's have a look at how your body does this. So here's the enzymatic step. catalyzed by pyruvate dehydrogenase. And just remember, the mechanisms of control that we're looking for here are completely linked to dietary status, whether you're well fed or whether you're starving. So the first points of regulation is when you get a buildup of NADH or acetyl-CoA, in effect feeding back and inhibiting pyruvate dehydrogenase. So this is a classic product inhibition scenario. Now, it may seem a little counterintuitive, but when you're starving, you'll actually have quite high levels of NADH and acetyl-CoA. The reason for this is that your body will be actively mobilizing fat. The process of breaking down fat is called beta oxidation, which we'll be telling you about soon in the fatty acid lectures. And beta oxidation releases a whole pile of NADH and acetyl-CoA. So in effect, fat mobilization and beta oxidation caused through starvation directly inhibits pyruvate dehydrogenase, in effect looking after that carbon as pyruvate, which is still in a glucogenic form, meaning we can make glucose out of it. The next form of regulation occurs through this enzyme's own regulatory subunits, pyruvate dehydrogenase kinase, which switches it off by phosphorylating it, and pyruvate dehydrogenase phosphatase, which switches it back on by removing that phosphate group again. So therefore, the mechanisms of control have to be linked to how we control these two regulatory subunits. And we do so through the action of these substrates, all of which increase in concentration during starvation. When you starve, you mobilize fat, and this is broken down in your cells via beta oxidation, releasing heaps of NADH, acetyl-CoA, and producing lots of ATP. These in turn activate pyruvate dehydrogenase kinase. This causes the phosphorylation of pyruvate dehydrogenase, switching it off. So in effect, starvation and fat mobilization ultimately stops you from converting a potentially glucogenic carbon source of pyruvate into a non-glucogenic source of carbon in acetyl-CoA, which of course is all completely linked to the energy status of your body. Now, to undo all of this, in the well-fed state, your body releases insulin. Insulin itself activates pyruvate dehydrogenase phosphatase, removing that phosphate group from pyruvate dehydrogenase, converting it back into an active form. And therefore, in a well-fed state, your cells will happily convert pyruvate into acetyl-CoA, either to be used in the TCA cycle or potentially to be stored as fat. So in conclusion, we've looked in depth at the regulation of glycolysis through its key rate-limiting steps and its irreversible steps, which are hexokinase, phosphofructokinase 1 or PFK1, and the last step of glycolysis, pyruvate kinase. And although not specifically a glycolytic enzyme, we've also looked at the regulation of pyruvate dehydrogenase. And this one, of course, is crucial because it's controlling the disposal of that carbon that has been processed through glycolysis, regulating whether it gets converted into a non-glucogenic form as acetyl-CoA or gets retained to potentially be turned back into glucose. We've looked at how all of this is controlled through the hormonal environment of the cell, which is closely linked with substrate availability through your diet. And although we haven't gone into it in too much detail just yet, we've looked at how your cells can regenerate NAD+ from NADH, doing so in an anaerobic scenario through a formation of lactic acid, or in an aerobic scenario through the use of electron shuttles and oxidative phosphorylation, which we'll cover in the next lecture. And just to help you recap this information presented in this lecture, here's a series of questions that you can ask yourself. These will help you to pull out the main themes from this material, giving you a deeper understanding of what we've covered.